My name is Victoria DeGrezia, and today I will be doing an experiment in order to test the effect of antacids on different weaker acids. So the weaker acids that I will be using in my experiment today would include orange juice, Pepsi, vinegar, wine, and then for my control group, I will be using water. So the purpose of conducting this experiment would be to see if the pH levels of these weaker acids would be affected if I drop a Tum in each of the different glasses that include the different liquids that I had just described. And the Tums will be the antacid that I will be using in this experiment. So one reason as to why I was interested in conducting this experiment is because I knew that antacids help to reduce or neutralize the effects of acidic foods or drinks that one can intake. So when I was discussing this topic with my aunt, I came to the conclusion that I actually wanted to do an experiment that would test this knowledge and kind of increase of my knowledge of what I knew about it. So I was very curious to see how an antacid would affect the levels of different weaker acids, such as the ones I just previously mentioned. And the reason as to why I selected the acidic liquids that I also mentioned is because these liquids are commonly used and included in many diets that humans have. So the orange juice, for example, is a very common acidic drink that we drink with breakfast. And for adults, wine would be another option. So these are the reasons why I selected those as opposed to other ones. To add, there are certain people who take antacids frequently in order to kind of help with gastrointestinal distress. So I'm conducting this experiment because I was wondering how effective it was as an over the counter readily available medication as opposed to something that is prescribed by a doctor. So my problem for this experiment is basically how will antacids affect the pH levels of weaker acids? For example, will they make the weaker acids less acidic or will the pH levels remain the same? I hypothesized that the Tums, which is the antacid that I will be using in this experiment, will make the weaker acids less acidic. So to expand upon this, I hypothesized that the Tums will be placed within the orange juice, the Pepsi, the wine, and the vinegar. And as they dissolve, these weaker acids will have a decrease in the hydrogen ion concentration as the antacid breaks down within each of these liquids. So the reason as to why I included water, even though it's not an acidic liquid, is because I wanted to have a control group. So I will still be placing a Tum within that glass as well and testing the pH level before and after. I believe that the purpose of an antacid is to serve as a neutralizing agent and it helps to lower the acidic levels within weaker acids. So I know it has been scientifically proven that baking soda can be used to reduce the acidic levels within someone's stomach. It's most often found with people who have like gastrointestinal upset. So by taking an antacid, that would help to increase the pH level, thus making it less acidic. So like I said, it is important to know that I will be conducting this experiment in 2 different intervals. So that means that I will, once I add the Tums in each of the cups, I will test the change or if there was any change in the pH levels after 5 minutes and then after 10 minutes. So I would be doing the experiment twice. So as for the methods, I will first make sure that I have all of the previous materials that I have listed, and that would include the measuring cup, the 5 different liquids. I will be pre-measuring each of the liquids just so I can wash the measuring cup between each liquid just to make sure that nothing is cross-contaminated and nothing would affect the results in the end. So those will all be pre-measured. I will make sure I have the 5 glasses, the 5 spoons. I'll have 15 test strips to first test the liquids in the first place before I do anything. Then after the first interval, use another 5, and then after the second interval, I would use another set of 5. So in total, that would be 15. And I would also make sure I would be using the measuring cup and anything else that I would need. So to begin, I would line the 5 different glass cups up in a line, and then I would take the pre-measured liquid and pour each liquid into a separate cup. So one would have water, one would have vinegar, one would have Pepsi, one would have orange juice, and one would have wine. And then before I do anything, I would take 5 of those test strips and test the pH levels of each of the liquids before I added the Tums or did anything to them. I would record that down and match that strip to the color chart matched on the side of the box. So I would have those results. Then I would take 5 different Tums and I would put 1 Tum in each of the cups. And once I do that, I will take a spoon, which is why I would have 5 spoons, and I would take a spoon and mix the Tum and that liquid together to make sure it's equally mixed, and I would do the same for each of the cups using a different spoon, once again to ensure that nothing was cross-contaminated and nothing affected the results in the end. Then once I do that, I will set the timer for 5 minutes, and I will wait the 5 minutes. After the 5 minutes are up, then I will take another set of 5 test strips. I will test the pH levels once again. I will record what the change was, if there was any, from the test strip to the box, and record that down on another chart. Then after I do that, I will wait another 5 minutes, so it would be a total of 10 minutes after I had placed the Tum and mixed them together with each of the liquids. So then I would take another set of 5 test strips and I would test the different liquids once again and see if the pH levels for some reason changed once again, and record those results as well. So the reason why I chose to two different intervals for this experiment is just to make sure that time necessarily didn't have an effect on the increase or decrease of pH levels. So I would just kind of wanted to make sure with that, and then also to increase the validity of the results. Okay, so now we're gonna start the experiment portion of this project. So I already kind of set up a lot of this stuff. So everything is pre-measured. So it's already a cup or 240 milliliters. So this would be the vinegar, then this would be the Pepsi or the soda, the orange juice, then wine or the sangria that I use, and I'll show you the brands later. And then this is just regular water. So I just pre-measured them all, that's why they're in these cups, and then I'm gonna pour them into each of the 5 cups. And like I said before, the reason why I did that is just to make sure that nothing is cross-contaminated and that won't have an influence on the results in the end. So to begin with, I'll pour the vinegar into the first cup. So that was 1 cup. And then I will pour the Pepsi or the soda into the second cup. And I made sure the cups were clear just so you could visually see what was going on. Then the orange juice in the third cup. Then the wine or the sangria into the 4th cup. And then finally the water into this cup. Okay, so as you can see, they're all pretty even. And now we'll move on to the next portions now that this is all set up correctly. So first, like I said, we're going to be testing the pH levels within each of the cups just to have a base record of what they are to begin with. So basically what you do is that you take the first strip, and like you can see there's 4 different little boxes, and then you just place it and make sure that all of the 4 boxes are in the liquid. So like I said, this would be the vinegar. So you're just gonna hold it for a second. And then you're gonna take it out, and you could see that it already started to change. So I'm just going to place it on the edge of this plate right here. And then I will do the same for the next one. So this is the Pepsi. So once again, make sure it's in the liquid. And then you could see it change a little bit. So then I'll put this on this plate as well, making sure that they don't touch, just so nothing is mixed. Then I will do the 3rd strip. So do it in the orange juice. And I will tell the results at the end. Okay, so then this is orange juice. I'll put that on the plate as well. Then we'll do the wine. Okay, you could see the results here. So far that was the darkest one out of all of them. And then finally the water. So just place it in the water. And then we'll put this on this plate. So these are the results from left to right. You can see this is from the vinegar, then this is the Pepsi, the orange juice, the wine, and then the water. So that's just an image of it now. Later on, I will give more details about what those results mean and compare it to the chart. So now that I did that, I'm going to take 5 of the Tums and put 1 Tum in each of the cups. Okay, so I'll just drop the first one in. And then I will do the second one, the third, the fourth, and the fifth. So now you can kind of start to see that they're dissolving. Okay, so like I said, I was gonna take 5 different spoons and just mix everything, but I'll do 1 per cup. Okay, that's the first one. This is the second one. And you can actually see this one's actually kind of fizzing as it mixes with the Tum. And this is the wine. And then finally the water. And they kind of look similar, but this is the wine and this is the water. I just put them on 2 different ends just so you can kind of tell them apart. Okay. So now I will be taking the next 5 strips and retesting them after the 5 minutes. So now we'll just wait, and then once that 5 minutes is up, we'll retest them and I'll show you that. Okay, so as we were waiting for the 5 minutes, it's going to be up in about 20 seconds, but I'll just go over the results from the initial. So basically for the vinegar, the initial acidic level was 3. This was the first one. Then the second one was the Pepsi. It was also a 3. Then for the orange juice, it was a 5. And then for the wine, it was 2. Just shut the timer off. And then for the water, it was 6. So obviously the water had the highest, which means that it has the lowest acidic level. And the highest acidic level was the wine, which was the 2. So now that the time is up, we will do, you can see that a lot of them are starting to fizz. So I'll just stir it around one more time. And then we'll take the levels again. And this would be after the 5 minutes. These are the results after that. So for the first test strip, once again, I'll put it in the vinegar. Okay. So I'm gonna put that down. Then I'm gonna do it for the Pepsi. I still have to compare the levels to the chart, but they already look like they're different colors, and it looks like they're kind of, if you look at the chart, I'll show you it later, it looks like they are the colors more like the less acidic levels. So now we're doing the orange juice. Now we'll do the wine. This one's still the darkest out of all of them, but remember that had the highest acidic level to begin with. and now we'll do the water, which is the final one. Okay. So now, once again, from left to right, it is the vinegar, the Pepsi, the orange soda, the wine, and the water. So if you want to look at them next to each other, so this plate was the initial, and then this was after the 5 minutes. So once again, I will now record the results and I will explain what they mean in a minute, but you can already see the change in color. So once again, initial, and then after the 5 minutes. So now we will be waiting another 5 minutes, and that will lead us to a total of 10 minutes after I place the Tums, so that will be the second interval. Okay, so we're still waiting for the next 5 minutes to finish up. So we have a little time, so I'll just go over what the levels changed to, and I'll kind of show you exactly how I did it. So for the vinegar, it was initially a 3, and then it became a 4. And then for the Pepsi, it was initially a 3, and then it became a 6. Then for the orange juice, it initially was a 5, and it remains a 5, so that one remained the same. For the wine, it was a 2, and then it became a 3. And then for the water, it was a 6, and then it became a 7. So all of them increased except for the orange juice, but the biggest, the one that had the biggest increase in the levels was the Pepsi. So that was after the 5 minutes. So now I'll just kind of explain how I did it. So the first one was vinegar. The second one was the Pepsi. Then the third was the orange juice. Then the fourth was the wine, and like I said, the fifth was the water. So I'll just take, since this had the biggest increase, I'll take the Pepsi and I'll explain how to do it. So this is the side of the pH level strip bottle. So basically you just compare it to each of the ones. So here you would compare it, so it was not necessarily a 5 because that looks a little bit more yellow. So then you go to the 6, and it kind of looks like that one. It looks very similar. If you go to the 7, just to see, you keep going just to see if it's too much or not. The second bar in my hand, it's more yellow, and on the thing it's more green, so this wouldn't be accurate. So you go back to the 6, so this one would read as a 6. So that's why I picked 6 for this one. So I just did that for each of the levels, and I just showed you that one because that was the biggest increase and it was very clear cut. So I did that for the initial set of strips, and then I did that for the one after 5 minutes. So now let's see how much longer we have on the timer. Oh, we actually have a few seconds. So then I will mix it again, and I will take another, I'll do another round of the test strips and test it again. Okay. So once again, I will just mix it. So like I said, make sure that all 4 bars are placed under the liquid that you're using, just so you make sure that it reached all of them, so it doesn't affect the end when you compare them. Just make sure they were all covered in the liquid, because if they're not, then it wouldn't change. So this was after the 10 minutes. So now I'll put this here. That was the first one. And if you see that the end of the strip is just a little like pink colored, it's just because of the Tums it mixed with the clear liquid, so that's why it looks like that. Okay, this is the next one. Then the orange juice. And the wine. And then finally the water. So now if I turn all of the plates, just so you can visually see it. So this was the initial results. This was after 5 minutes. And then now this is the one we just did, and this was after 10 minutes. So from left to right, once again, it's the vinegar, it's the Pepsi, the orange juice, and the wine, and then the water. So now I will just compare the final results with the side of the bottle once again, similarly to just how I showed you, and I'll record them down, and then I'll go over what they are. Okay, so now I compared the test strips after the 10 minutes to the side of the bottle, like I said, how I showed you, and I recorded them on the paper. So for vinegar, the first test strip, it went up one more level. So it was a 4 after the 5 minutes, and then after the 10 minutes, it became a 5. So the color just slightly changed, but it did increase a little bit. Then for the Pepsi, it remained the same. So it was a 6 after the 5 minutes, and after the 10 minutes, it remained the 6. So the color was the same. Then for the orange juice, once again, it did not change. So it was a 5 throughout this entire experiment. Then for the wine, it also remained the same from the 5 minutes. So at 5 minutes, it was 3. And after the 10 minutes, it was also 3. Then for the water, after the 5 minutes, it was 7. And after the 10 minutes, it was also 7. So basically the only one changed after the 10 minutes was the vinegar, which was, it went from a 4 to a 5. So before we did the experiment, just to kind of put all the liquids in levels based on their acidic levels. So the least acidic was the water, and then the next least acidic was the orange juice, and then following that was the Pepsi and the Pepsi and the vinegar. They were tied, and then the wine was the most acidic. And then after we did the experiment and we added the Tums and recording all the results, now I'll tell you the order of what it is. So the least acidic was still water. Then the next one changed, so now it would be the Pepsi. Then after that, it is the orange juice. Then it is the vinegar. And then it's the wine. So from the beginning to the end, the wine was the most acidic, so this stayed the most acidic, and this stayed the least acidic. So I realized that after doing the 2 different intervals, so I did the 5 and then the 10, that only the one increased, the vinegar increased from the 4 to the 5, but in general, as a collective whole of all of the liquids, they didn't really change with the extra 5 minutes. So based on that, I don't necessarily believe that it was the time that would increase the acidic levels. I think it was just based on the liquid and whether it became less acidic when it was mixed with the Tums. So basically that is it for this experiment. I will now explain a little bit more about the results, but this is how we conducted it. Okay, so something just to keep in mind is as I go over the results and what happened throughout the experiment, I will be saying numbers. So in case you don't know what that means, I'm just gonna kind of go over it now, just so you have an understanding of what I'm talking about. So basically, there is a scale from 1 to 14, and that is a representation of how acidic a liquid or something is. So when I say a liquid went from a 5 to a 6, the increase in number means that it is less acidic. So the closer the number is to 1, that would mean that it's more acidic. And then the closer it is to 14, that would mean that it's less acidic. So as the numbers increase, so when I'm saying that it goes from one number and that number jumped and went up more pH levels and that number increased, that means that that liquid became less acidic. If the number stayed the same, that means that the acidic level remained the same and the pH level didn't change. But nothing in this experiment went the other way, meaning that it didn't necessarily decrease or become more acidic, because the point of the experiment was to see the effect of antacids on weaker acids. And basically what would come out of that is that the antacid would decrease the hydrogen ion concentration within each of the weaker acids, thus making it less acidic. So that's just something to keep in mind and something I wanted to go over just to make sure that you guys know what I'm talking about. So now I'm basically just going to go over the results from the experiment and just kind of wrap it all up. So to reiterate, vinegar initially had a pH level of 3. After 5 minutes, it was 4. And then after 10 minutes, it was 5. So it increased by 2 levels from the beginning to the end of the experiment. Then for Pepsi, it initially was 3. And then after 5 minutes, it was 6. And then after 10, it still remained the 6. So this had the biggest increase out of all of the liquids because it went from a 3 to a 6 and it jumped 3 levels. For orange juice, it remains a 5 throughout the entire experiment, so it did not show a change at all, even after the antacid was added to it. Then for wine, it initially had a pH level of 2, and then it went up to a 3 after 5 minutes, and that remained the same after 10 minutes. It was also a 3, so it only increased by 1, but it still did show a change. And then for water, it initially was 6, and after 5, and even after the 10 minutes, it had the same, so it was a 7. So it went from a 6 to a 7 for water, so that one also increased by 1 as well. So before I did the experiment, I basically kind of put all the liquids in order from least acidic to most acidic. So for least acidic, it was water, then it was orange juice, and then Pepsi and vinegar were tied. It had the same level, and then wine. Then after the experiment, after going over the results I just have, that I just went over, it is now water, and then it's Pepsi, orange juice, vinegar, and wine. So 2 similarities from the beginning to the end. So water remained the least acidic and wine remained the most acidic. So even though there was changes in all of them, those 2 did kind of remain in the same spots when you kind of put them in order from least to most. So that's just some things to keep in mind. But the orange juice did not show change at all. So that's a key takeaway from this experiment. And another one would be that the Pepsi showed the biggest increase. So now that I talked about the results and I talked about what happened throughout the experiment, I'm just kind of reflect back on my hypothesis and from the beginning of the experiment before anything was done. So basically, my hypothesis was correct. I predicted that after adding the Tums, which was the antacid that I used in each of the weaker acids, the acids would become less acidic, and that's basically what happened, as you can see through the results, where the numbers pretty much, with the exception of orange juice, increased. So that means that as they became closer to 14, or the numbers increased, that means that the liquid became less acidic. I did not expect Pepsi to have the biggest jump in their pH levels. So initially, Pepsi had a level of 3, and after the experiment was over, it ended on a pH level of 6, so it jumped 3 levels. Another thing that I did not expect was for orange juice not to have a change at all in their pH level. So throughout the experiment, it remains a 5 regardless of whether there was antacid mixed in or not. So from beginning to end, it was 5, and there was no change in that result. So those are 2 things that I kind of just wanted to point out because they were 2 things that kind of stuck out to me and something I didn't necessarily expect. One thing like I want to just reiterate is that when the Tum was added in each of the weaker acids, that Tum was an antacid. It broke down and it dissolved. You can actually see it in one of the videos dissolving. I believe it was in the vinegar. And as it dissolved, it changed the hydrogen ion concentration, and that made the liquids less acidic. So that was basically what was going on throughout the experiment with each of the liquids. But that was with the exception of orange juice, because as I said, that acidic level did not change. So that, once again, that was something that I did not expect out of this experiment, and it is something that I would like to do a little bit more research on and find out more information, considering it was the only liquid that didn't show change, which I thought was a little bit interesting. So I guess from further on, I would like to have more knowledge about why this was the fact only for orange juice. So now I'm going to be talking about the future directions that I would take, also discussing what I would do different or what I noticed that I would change if I did the experiment again, or if I tried to um, increase the validity of the results. So one thing is that I noticed that the liquids that I chose were different colors. So obviously the orange juice was orange and the wine was like a deep red color. So those are just 2 colors that kind of really stuck out to me, because the water and the vinegar were just clear, and the Pepsi was also a darker color. That's also important to keep in mind. So my thing is that after doing the experiment, I wonder if the color of the liquid had an effect on the results of the experiment, because basically what happened during the experiment is that after I added the antacid, I dipped the pH strip in each of the liquids, and I would test that color of the strip against the side of the bottle that came with the pH strips, and I would try to figure out what level it was at after the antacid was added. So basically, I was comparing the colors from before and after. So what I am thinking about is whether the different color liquids had an influence on the color that showed up on the strip, hence influencing what number I selected when I compared it to the chart. So that was something that I would keep in mind and want to do a little bit more research if I did the experiment again, to kind of make sure that the results weren't tainted, and if they were, how to resolve that. And then just something to keep in mind is that with this knowledge, people should make wiser choices with their dietary intake of foods and liquids, especially if they're prone to having gastrointestinal distress. Another important thing to keep in mind is that proteins and enzymes have a very important function within our body, and this is because enzymes tend to be very specific within the pH range that they work in. So altering this range within the body is basically what contributes to issues, and that's basically what antacids are for. Their goal is basically to try to decrease the distress or decrease the acidic levels within the body or within something that you're talking about, in order to kind of resolve it. So that was basically what I was testing in this experiment. I was trying to see whether antacids such as the Tums would decrease the acidic levels of the different liquids that I used. That would include the water, the Pepsi, the orange juice, the vinegar, and the wine. So I tested all 5 liquids, and as a whole, all of them pretty much change except orange juice. And now looking back at the experiment, I see that my hypothesis was correct, but in regards to future directions, I would like to kind of monitor what I was doing a little bit more carefully, just to make sure that, like I said, the color of the liquid didn't have an effect on it. But as a whole, I thought that the experiment was very successful, and I hope you enjoyed it.